I've been at Hopkins for about, since actually beginning of 2013, so it's about 12 years. So my lab mainly focuses on the development of new technologies for molecular analysis of diseases. I have a lot of overlap in terms of interest with a lot of faculty here. And today I'm going to tell you some of the technology that we have developed and used to achieve the highly sensitive detection of biomarkers for cancers and also for infectious diseases. So I will begin with this slide that we are interested in looking at the genome. So as we know that human genome contains numerous alterations at different levels at chromosome, and also nucleotides. This includes irreversible changes in DNA sequences such as genetic mutation. Also include the change of DNA genes or copy number variation. This also includes the change in the structure as well as potentially reversible changes, also known as epigenetic modification, such as DNA methylation or histone modification. The study of these genetic and epigenetic changes has now become a very active research areas in modern medicines, because the study of these aberrations, molecular aberrations, can provide the clues to the mechanisms of disease pathogenesis. And from the application side, that we can use these studies to identify molecular events that can be used for targeted delivery. And so in my lab, we are more interested in using these molecular aberrations to facilitate the use and discovery of different biomarkers in a wide range of disease managements, including early detection. I will give you some examples. Diagnosis, prognosis, and also to select the proper treatment plan for specific patients, even though the patients have the same diseases, but the genome profile is different. They will have different outcomes, and we can also use these markers to facilitate post-therapeutic monitorings. While the use of these genetic and epigenetic markers has been shown to have good promise, very good promise, but currently the utilizations of these genetic and epigenetic markers are still limited mainly to research laboratory or clinical laboratory, but not yet in routine clinical settings. There are several technical barriers that prevent the widespread adoption of the use of genetic or epigenetic biomarkers in routine clinics. One is that it has the current methods does not have sufficient sensitivity and specificity, in particular for some challenging samples. For example, like our lab, we are interested in cancer detection. So the challenging sample, liquid biopsy is right now people treat as holy grail. You want to detect the change cancer-specific markers in blood, serum, sputum, or urines. And current technology typically just do not have the level of sensitivity and specificity sufficient enough to handle these challenging samples. And also the current technology, including microarray, PCR, even the next-gen sequencing, all involve the tedious sample preparation process. This also makes it quite difficult to be routinely used in clinical settings. And cost, of course, is not an issue. Give you one example. For example, currently there are 2 FDA-approved genetic tests for breast cancers, including Mammoprint and also Oncology DX. So each test for this FDA-approved, it costs $4,000 per test to get these genetic profiles for these specific cancers. Micro and nano technologies, I'm going to give you some examples in microfluidics and also the optic technologies that we developed, shows high promise to tackle these challenges. For example, we know that the material properties at a small scale down to the nano or micro scale has a significant change in their properties, and such as the gold metallic particles and also semiconductor particles has a superior optical properties that can be used to enhance the sensitivity. And here, a lot of faculty colleagues here working on microfabrication. We know miniaturizations of assays and technology and devices through microfabrication or even nanofabrication allow us to reduce the consumptions of reagents and then reduce the cost, and also allow us to parallel processing to enhance the throughputs. And here, the device eventually can also be used for point of care because of the cost reduction and also because of portable natures, as a lot of activities are going here. So this we'll be able to use to address the aforementioned challenges in using the genetic and epigenetic markers in routine clinicals. So I'm going to give you some examples. The first example I'm going to share with you is the use of optics, try to enhance the sensitivity of biomarker detection. So in my lab, we use confocal optics to do the biomarker detection. So these general concepts of confocal. Using the confocal optics, we confine the detection to be a small detection volume in the range of femtoliter size by its light diffraction limits and also by the pinhole detection. So with this, we know if we are able to detect a fluorescent molecule within this small detection volume, this volume can substantially suppress the background noise coming from scattering, coming from the impurities. In the end, we are able to detect the signal coming from even single molecule, like this is the single molecule trace. This is only single fluorescent dyes. You can see this peaks represents a single molecule event and represents the molecule passed through the detection volume where the molecule gets excited by the laser and emits a fluorescence at a different wavelengths. And you can count the number of fluorescent molecules by counting directly the number of the peaks. And the bandwidth, the peak width of this peak represents the time that the molecules stay in this detection volume can be used to determine the mobility and also binding events of these molecules. So provides a fruitful of kinetic information that can be used to do a lot of studies, including sensing detection. One example is the detection is this concept of two-color coincidence detection. So if we like to detect a specific target, can be a protein target, can be DNA targets, you can use two probes. These two probes can bind to this target simultaneously. But these two probes are labeled with different fluorescent tags, green or red in this case. So if we have the single molecule optics, we can measure this the detection directly in a solution without the need for separation. Most assays are ready. You need to separate, remove the unbound probes. By using this technique, in this case, let's say if you have specific targets, these two probes can bind to these targets, and then we can detect simultaneously the signal emitted from two single molecules, these dye molecules, red and green molecules. And these coincidence events can be used to determine presence of targets. In contrast, if the sample does not contain specific targets, these two probes will still be measured by the optics. And you see these single molecule events, but they are random signal. They are not synchronized. So using this coincidence event, you can provide the evidence to determine the presence of this target and also to quantify the targets. So I'm going to show you. So here's the experiments that that was done using the quantum dots and labels. We labeled the two oligonucleotide probes specific to these DNA targets. One with green quantum dot, the other one with red quantum dot. The advantage of using quantum dot, multiple one, but one is that you can use a single wavelength to excite the different color of quantum dots. So in this case, that you see, we are able to detect the specific targets in this positive control here. You see a lot of the coincidence events. Again, so here I forgot to mention this is measured using single photon arranged photodiodes with a megahertz, the frequency, namely that you have a microsecond temporal resolution to confirm the coincidence events. This allows us to be able to detect specific DNA targets directly in solution without the need to separate unbound targets. So we can also expand these assays for markers. In this case, we are interested in genetic mutation markers for cancer. So if we were to detect the genetic mutation, we can couple the coincidence detection principle with DNA ligation reactions. So if you design the probe, again, similarly, a pair of probes specific to a target, and it has specific target but with one mismatch here. So look at the perfect match case. If we put enzymes to the solution, the enzyme will fix these link points, will link these two probes together using these perfect match targets as templates. And then we can denature these duplicates and using quantum dot to capture this ligation product. Then we are able to detect the coincidence emissions found the quantum dot as well as found this green dye there, and we can detect that this particular genotype or this is. But if this is the mutated target, even though you have only one point mutation, there point one nucleotide which is not matched, and this mismatch will prevent the DNA ligase to link these two oligos. So quantum dot will only capture this capture probe, and the fluorescent dye on the other probe is away from the quantum dot, so you don't see coincident events. So you will be able to, based on this single molecule coincident detection, to detect the mutation. And here it just shows that this is the ovarian cancer of the patient samples that we monitor. We detect the KRAS codon 12. This is one of the important hotspot, KRAS codon 12 for ovarian cancer, so that we can detect this directly from the patient samples. You can also expand this coincident detection to multiplex detection by use combination analysis of the two-color coincident detection. For example, we can design a set of 5 primers, and we said 2 primers specific to one is to mutant target, the other one is to wild type targets, and then we can generate the target specific oligonucleotides, ligation products, and then use quantum dot to capture these ligation products. So if the sample contains only the homogeneous mutants, the targets, we will see 2-color coincidence only between the red colors and blue colors. If the sample contains only homogeneous wild types, we will see the coincidence emission between red and green colors. If this sample contains heterologous targets, then you see the coincidence in 3 colors. But this allows us to be able to expand this concept to analyze the multiple targets or different polymorphisms. And so I'm going to show you another concept that you can also use the single molecule detection. Try to detect specific target based on the mobility shift upon binding. So in this experiments, we use the labeled DNA probe to try to search for the specific targets. And we can measure in the absence of targets. We will again measure the single molecule signals, single fluorophore signal. You see a peak here, and has a characteristic time constant tau1 here. But if we measure the sample contains a specific DNA target, bind to the target, probe bind to the targets, cause the mass change and mobility shift. The mobility will become smaller. So you expect that you can detect the DNA again, the fluorescent dye, but you will measure the fluorescent signal with extended time constants. So this time change or mobility shift can be used to determine the presence of targets without, again, the need to separate the unbound probes. So this is the concept of fluorescent correlation spectroscopy. So here I show that this is the control, negative control. You only have the molecular probe without the presence of targets. We see a lot of single molecule signals. And this is the positive control. You see, again, single molecule signals. But if you analyze, determine what is the time constants associated with these peaks, you do autocorrelation. You found that for the the unbound probe, it has a time constant about 200 microseconds, 200 microseconds. But upon binding to the targets, the time constant shift from 200 microseconds to about 500 microseconds. So this shift can be used to determine, again, the presence of target or sensing. And you can also quantify the different level of targets by the different levels of time constant shift using this technique to do it. And this technique can be also extended to do different binding. In this case, we are examining the binding between protein, DNA, and also cancer drugs. So we look at different drugs, in this case, M4M drug and also doxorubicin. So this drug can bind to the DNA to inhibit the transcription factor bound to the DNA, and a gene can be silenced. So we're actually seeing how this binding is working and what is the level of binding. So using this technique is that we have labeled DNA, and then we can determine, we can characterize the intrinsic DNA, the fluorescent signals. And then if the protein binds to the DNA, we can measure the signals, and this signal has an extended time constant because of the mass change. And if the drug binds to the DNA, kick off these proteins, then the time constant will go back to the smaller regions because the protein is not associated with the DNA anymore. So by measuring this time constant change, we can know what is the binding states of these targets for particular promoter sequence and also different drugs and different concentrations. So we can look at this one to monitor the binding kinetics of the drugs to the DNA. I highlight that we are interested in using the confocal optics to enhance the detection sensitivity. But I would also like to highlight that there are some intrinsic problems associated with the use of this confocal optics for detection. One major problem is that it has a very low mass detection efficiency, because the high sensitivity only occurs when the molecule is right in this detection volume. But you need to have a way to be able to shuttle the molecules right to this detection volume. Using microchannel is an approach, but again, this microchannel typically, if you're talking about a few tens of microns, is much larger than the detection volume here is about 1 micron or some micron. You can use a narrow channel to shuttle your molecule to make sure you have 100% detection efficiency. But narrow channel is just difficult to handle larger volumes, and you always have clogging issue there. So what we have done that in the past, we have been using it as a kinetic, try to do the focusing to deliver the molecules. Today I'm not sure I have this. So I don't have slides talking about this in so kinetics way to overcome this problem. But I'm going to show you one of these modified confocal optics design that we have proposed a couple years ago. It's called cylindrical illumination confocal spectroscopy, or KICS. So again, I'm using this slides to show you the difference between the standard single molecule confocal spectroscopy and this new design. In the standard confocal spectroscopy, the laser light is tightly focused in the two-dimensional, so diffraction limits. By using, in the new design, we perform one-dimensional relaxation. We performed, using cylindrical lens, try to create a sheath-like illumination volumes. In the vertical direction, is still diffraction limits to make sure we have a small observation volume to minimize the background levels. But on the lateral direction, we expand it to become like a sheet. And then the second is that we use microfabrication to fabricate a rectangular aperture to collect the photons only within these regions. And this region is more uniform because it's still Gaussian profile. And then we want to collect the illumination intensity that is quite uniform near the centers. And the third is that we use a microchannel to shuttle the micron. This is a microchannel. And using this microchannel to match with the size of this aperture and to shuttle the molecule through the detection volume. By doing this, we can make sure that every single molecule will be detected using this confocal optics and still with the minimal backgrounds, be able to detect a single molecule but with enhanced detection volumes. So we have applied this modified the confocal optics for different detections and for different biomarkers analysis. And here I'm going to only give you one example, is that we use these optics to analyze the DNA integrity biomarkers. So we are interested in the circulating DNA integrity. We know human serum plasma contain not only protein, it has a DNA, cell-free DNA, circulating DNA. This DNA coming mainly from the normal tissue, healthy cells. But some of this DNA is coming from tumor cells, if this is the serum from the tumor patient, cancer patients. So if the DNA is coming from the healthy cells, the DNA the cell died through programmed death, through apoptosis. The DNA wrapped on the chromosome got chewed up into small fragments. It's quite uniform, about 200 base pairs. But if this DNA is coming from the cancer cell does not go through apoptosis, programmed death. It's through the necrotic pathway. So DNA got chewed up in random sizes. So this has been shown that by analyzing the size distribution of a circulating DNA, it can be potentially used as a marker for cancer. So other groups, including immunology group at Hopkins, have demonstrated that in this case that more than 120 patients' sample got analyzed. They found that using PCR to analyze whether or not there exists larger DNA, and this is associated with cancer. They analyzed, they find high correlation that the cancer patients has higher chances to have a higher abundance of larger DNA, larger than 400 base pairs in the serum or plasma samples. Currently, so this suggests that the DNA integrity, namely the presence of the larger size of DNA in the serum plasma, can be used as a marker for pre-screening of cancers. And I want to tell you what is the results is done currently. Because circulating DNA exists in a very, very low physiological concentration, typically only a few tenths of nanograms per ml, which is equivalent to only a few thousands or a few tens of thousands of genomic equivalents per ml in the serum plasma samples. So it's very challenging to analyze this. People want to analyze circulating DNA using PCR to do the amplification. And in order to analyze the DNA integrity, the size distribution, so what has been done currently is that they will design two PCR primers. One will and select a specific loci or gene, housekeeping gene like beta-actin. And they have two sets of primers. One amplifies the sequence of about 200 base pair. So as I mentioned to you, for most of circulating DNA, it's about 200 base pair in size for normal cells. And this can be amplified. And then they have another pair which amplifies the larger fragments forming base pair. So because this is designed to amplify larger fragments, if these serum samples from normal patients does not contain larger DNA fragments, amplification will not occur, will not amplify. So then by comparing the ratio of the amplification for the larger DNA amplicants, smaller DNA, we can define this DNA integrity index, and this can be used to determine whether or not there's a high abundance of larger DNA in the sample. So this is typical way that people use to determine the longer DNA in the plasma samples. By using PCR, there are some drawbacks. For one, our goal is to analyze what is the overall distribution of longer DNA versus the smaller DNA. But you think about PCR can only amplify a specific loci. And you want to use the loci to represent the overall population of longer DNA and smaller DNA. It has sampling issues, right? It could be biased errors. And also is that PCR has a very limited sizing resolution. In this case, you only can analyze 200 base pair versus 400 base pair. You cannot analyze 300 base pair, 600 base pair, 800 base pair. Then why choose 400 base pair? This is just random choice, right? So it doesn't have a sizing resolution. Another thing is like we all know, you want to analyze DNA, you got to go through sample preparation process. When you go through sample preparation process using column or beads, sometimes it will cause DNA shearing. It happens because DNA shearing. So if you shear the DNA through the sample preparation process, the DNA integrity that you are going to determine may not reflect the actual DNA integrity in these patients. So we propose to use the single molecule method to analyze DNA integrity. So in this example, I'm going to show you here is that we can label the DNA with DNA integration dye. The number of dye bound to the DNA is proportional to the size of DNA. So the principle is that if we can measure the DNA, single molecule DNA intensity, then we may be able to correlate the intensity to the size of DNA. If it is true, then we are able to analyze the DNA size in low concentration serum plasma samples directly without using PCR, and we can analyze every single DNA, not a specific gene or loci. So the validation, this actually is the validation experiments that we analyze the DNA layers, DNA digest, and DNA separations, and then we are able to show that the number of photons, the burst size basically is the intensity of the DNAs, linearly correlate to the length of DNA as we would like to see, because the number of dye bound to DNA is proportional to length. So this gives us the encouraging results, allowing us to be able to analyze the patient samples. So here is the results of the integrity analysis directly from lung cancer patients. So this representative result here shows the comparison between the early stage and late stage cancer patients. And this is the first time that people are able to show the whole size spectrums of circulating DNA in serum plasma samples. And as you can see from this result here, you see here, if you look at the small DNA fragments, in fact, you don't see much difference between the late stage and early stage patients. But when you look at the larger DNA parts here, you see the late stage patients has higher abundance of the larger DNA than the late-stage patients. So this allowing us to define a more precise DNA integrity index and to be used as a pre-screening, not very specific, but it is pre-screening of cancers, and also to be able to monitor tumor burden. You have different tumor burden after the surgery. You want to see when I relapse, can be used to monitor the tumor burden because numbers of the large DNA is supposed to be proportional to the size of tumor in the patients. So we show that we are able to use a single molecule method to analyze DNA integrity, and we show that we only conduct a small cohort of patient samples, and we show consistently that the late-stage DNA, late-stage patients has a higher the integrity index than the early-stage cancer patients, and even and for the non-cancer patients, the index is typically lower than the cancer patients, as what has been reported previously using the PCR methods. Now I'm going to tell you that we can also couple these single molecule optics together with hydrodynamic separation technique, allowing us to be able to analyze or to size separation of DNA with very high sensitivity or using a only small quantity of DNA. So I'm going to introduce this concept of hydrodynamic separation. So if we were, so in this case, what we did is that we inject a small amount of DNA into a microcapillary. And this microcapillary, in fact, the inner diameter is very small, it's only 1 micron or some micron. And then within this microcapillary, if there are different particles or molecules inside this capillary, in this case DNA, the average velocity will be different. You have this the hyperbolic profile. And normally when you have a particle inside this capillary, through the hydrodynamic ratio to go through this capillary, you have this average velocity. But if the channel is so small, smaller to really comparable to the molecular sizes, the size exclusion effects occurs. If you compare smaller and larger DNA molecule, larger DNA molecule will be more excluded from the side walls. So in the end, you will calculate the average velocity. So the larger molecule, because it's relatively more away from the side wall, so it has more near the center, so you have a higher average velocity than the smaller DNA. So over time, this will allow us a way to be able to separate the DNA by their sizes. So this is the results of DNA separations. We separate 7 different DNA fragments. This is the DNA latest from the New England Bios, and this is the one you can download from the website. Typically, you use this DNA later, and you want to see the different sizes, what's the DNA later, and you just do the gel electrophoresis. And I want to show you here, we can duplicate these results you can see from the capillary, from the gel electrophoresis, or even capillary electrophoresis. I want to highlight the key difference here. Let's typically want to do this kind of results. You need to use about a microgram or some microgram of DNA to get these laters. In this case here, we using single molecule separations. Here we only using femtograms of DNA. Order make it smaller than the DNA and be able to get very clean and results. And more interesting thing that I want to point out here, this class of DNA, in fact, this is a class of many single molecule DNA here. So if you look at this first picture, in fact, there are only 9 molecules. So here shows that only a few hundreds of molecules needed to do these separations. For the commercial DNA cell line, which is important, but if you're talking about clinical samples, this could be an effective way for us to analyze that the sample contain limited amount of DNA samples. So it's I guess it's very effective way to analyze the separation and also detection of DNA. So I'm going to switch gear to talking about the second technology. I have until 45. You say 45 minutes? Yeah. I guess I just got to run faster. So yeah. So here I'm going to show you the second technology, but I'm going to run a little bit faster. So here I'm going to show you the quantum dot technology. So we a few years ago we developed a technology called quantum dot phase sensor, DNA sensor here. The basic concept that we created this quantum dot nanosensor, it comprises of 3 elements, including quantum dot, which serve as the energy donor, and we have a pair of oligos and one the DNA probe, one label with Cy5, which serve as energy acceptor, and so biotin layer. Quantum dot is functionalized with everything. So the idea is that if we have the sample containing the targets, these two probes again will bind to the targets, and the whole thing will be captured by the quantum dot, and we can detect this narrow assembly or the presence of target according to the free emission, because the energy excited to the quantum dot will be transferred to the Tza-Huei dye here, if it is within 10 nanometers ranges. And then you can detect this by detecting emission of Tza-Huei dye here. So Frey is a very powerful tool commonly used. The people use it as a molecular ruler to monitor the molecular bindings. But a lot of people also using Frey to serve as a sensor to measure this binding. And if you would like to design a perfect biosensor based on Frey, and you need a perfect pair of the dyes to be used. In this ideal case, you need to fulfill 3 basic requirements. You need to find the dye, the flow for donors. The emission has overlap with the absorption of the acceptors. Make sure there will be energy transfer, for one. Secondly, you want to make sure that the donor emission and the acceptor emission has minimal overlapped crosstalk. Otherwise, you cannot differentiate these two. And you want to make sure, the third, that the energy wavelength you use to excite the donor need to be away from the absorption spectrum of the acceptor. Otherwise, you will direct excite the acceptor. So what you see here is not through energy transfer, means like it's not through binding. But in the real world, it's almost impossible to find a pair of dye that can simultaneously fulfill these 3 requirements. That's because of a traditional dye has a very small stock shift, the peak between emission and absorption, and has a very wide emission spectrum. So if you find a dye that has an overlap between absorption and emission, you have this crosstalk. And if you select a wavelength to excite effectively the acceptor, the donor, because of the short stock shift, you will also cannot avoid to certain level of direct excitation. This is commonly seen for the organic dye. By using quantum dye, you can overcome this problem, because quantum dye, as you know, the emission is size tunable. You can, and it has a narrow emission bandwidth. So you can tune the quantum dye that has overlap here, and you can excite the quantum dye with a wavelength far away from the acceptor, because the quantum dye has very wide stock shift. You can even using UV light to excite the quantum dye, which will not excite the dye chosen here. So you can minimize these 2 problems and to make this sensor to have very low background noise. So with this, we can enhance the sensitivity. What I'm going to show you here is that the result here. This actually is the quantum dot negative control and positive control. This again is monitored by the highly sensitive single molecule spectroscopy. So you can see here is like in negative control, you don't have DNA target. You see very low background noise because of the advantage I mentioned to you. But about detection, when the positive control, you have the targets, you can detect the single quantum dot at single dot or single molecule levels, show the phase signals, and with very high signal to noise ratio at single particle level. And we have using this quantum dot phase sensor for different detection, genetic and mutation and copy numbers. Now I'm going to show you the example of using this one for DNA methylation epigenetics. So my lab has been working with different groups epigenetics at Hopkins for a couple years. So one thing is that we are interested in DNA methylation detection. DNA methylation is contributes to the progression of tumorigenesis by silencing the tumor suppressor gene. It has been known that if it is DNA coming from the normal cells, the promoter of tumor suppressor gene is typically unmethylated. But if the DNA coming from tumor cells, the promoter of tumor suppressor gene is often hypermethylated. And because methylation will cause the structural change of chromosome to become much more compact to prevent the protein binding to the DNA. So this silences the whole gene. And so DNA methylation can be used as a marker for cancer. But more importantly, more recently, our groups, we and also other groups have shown that DNA methylation is early events and can be detected even from tumor precursor. So we have shown that DNA methylation can be detected from polyp precursor for colon cancer. Last year, we just published a paper. We show that DNA methylation can be detected in tannin, which is a precursor for pancreatic cancer. We know there's no marker currently available for pancreatic cancers. So this suggests that DNA methylation not only can be used as a marker for cancer, but can potentially be used as a marker for early detection. So the current method for DNA methylation detection is MSV methylation-based PCR, developed by Jim Herman and Steve Badins, my colleagues at Hopkins here. So these are the gold standard or most widely used method for DNA methylation detection. In fact, there are companies already selling the diagnostic kits based on this. In Belgium, there are 2 companies using these methods. And this method has a very high specificity, can detect one methylated DNA in a background, high background, about 1,000 of unmethylated DNA. But the sensitivity of MSP so far still not good enough to reliably detect challenging samples. I mentioned earlier, serum plasma, circulating DNA, tumor-specific DNA, sputum, or even urine or stool for colon cancer. It just does not have a level of sensitivity to direct detection. So together with Jim Herman and Steve Badins groups, that we developed a technology called MSQ methylation-specific quantum free technology. Basically, we combine the high specificity of MSP technology together with the high sensitivity of quantum free technology to do DNA methylation detection. We first conduct bisulfite conversion to convert the unmethylated citizen to become uracil. So by doing this chemical treatment, initially the methylated DNA and unmethylated DNA are genetically identical. Sequences are same. But with this bisulfite conversion, we convert it to the DNA sequences of different strands of different sequences. Then we can using PCR primers or using quantum dots start to detect this methylated DNA. So using this technique, we show that the level of DNA methylation, you can see with the increase of DNA methylation, you see the increase of the sci-fi signal, energy acceptor signal, but the decrease of the quantum free signal. And we can quantify the level of DNA methylation by the level of energy transfer. And we also show that we are able to quantify. This is an example showing that we using this quantify to monitor the patient responses upon the epigenetic treatment. We look at this MDS patients, which is pre-leukemia patients, and this patient has been undergoing the epigenetic treatment with azacitidine, which is FDA approved, the epigenetic drugs. And then we show with this method, we are able to monitor the patient responses to the drugs very often. Every other week, we can monitor it. As opposed to that, you have to ask patient to come back 6 months to see what's the outcomes of your. So we show that with this one, we are able to monitor the patient responses. And then not every patient response is what we expect to see the change of DNA methylation. This also highlight the importance of personalized medicine. If we are able to measure response of the patient upon specific treatments and know someone just work well, the other one doesn't work well, then it's time that we have to tell our clinician to change the treatment plans because it's not going to work for these particular individuals. So with this method, we are able to do frequent monitoring, be able to provide this information to the clinicians. I want to go to show that this method just has a very high sensitivity. We show that we are able to detect about 15 picograms of DNA, about 5 copies of genomic equivalence in a very high background of healthy DNA, about 10,000 backgrounds. And this is very interesting result. This is from heavy smoker. We are analyzing whether or not we can see the DNA methylation in sputum samples from the patient. They are not lung cancer patients. They are heavy smokers, 50 years male and 50 years old. We analyzed the 20 patient non-cancer the sputum samples using gold standard MSP method. We did not detect any methylation in the sputum samples. Only when we do 2-stage nested MSP, we see 3 patients, patient 3, 7, 8. This patient has TMS1 contain methylation. These are extremely high risk subpopulations from these heavy smokers that there is a high chance of developing lung cancers. So this is very important information for us to know. Unfortunately, this result has been obtained using nested PCR. Very tedious process. Is totally clinically non-user-friendly methods to do this. We show that with the quantum dots method, we can identify these 3 patients with very high signal to noise without the use of nested amplification. So I'm going to show you another. I guess I will skip this one in the interest of. So So I'm going to show you, switch the third gears on this, the microfluidic droplet technology here. So, so my lab, we are also interested working on the microfluidic technology, particularly in droplets. And I'm going to show you two different types of a droplet technology formats. So the first one is this the two-phase flow droplets. You can using this cross channels and use side sharing to generate the droplets with high frequency, about 1,000 hertz, and the size can be nanoliter to picoliter sized. So we have been using these droplets as a reactor for a lot of applications. I'm going to show you some of So using this one, you can make it very high throughput, means like you can generate millions of droplets within 1 hour, using this as reactor to do a lot of interesting experiments. And then we also show that, before I show you the example, I also want to show you that we can couple our single molecule counting technology together with the droplet technology. So we are able to count each individual droplets and the numbers of genetic contents or RNA or DNA trapped within these droplets. So this can be the single cell and release the DNA or RNA within this. So we are able to quantify this using this integration of the optics and also the droplets. Now I'm going to show you one example of digital analysis of pathogen here. So here I'm going to show you that just a lot of people here working on infectious disease. You know that if you want to do detection, specific detection, special genetic detection, you got to use a PCR because the concentration of pathogen that you can get from the sample is so low. So give one example. If you have one bacteria in 10 microliters of samples, you have only a few copies of DNA, a few thousand copies of RNA, automolar or femtomolar. This level is just not possible that you can do direct detection. You got to do PCR amplification. But if we are able to, using a technology to divide this 10 microliter into millions of droplets, each one is only 10 picoliters. And then the bacteria will be only trapped within one droplet. So if you do this, if you lyse the bacteria, DNA released from the cells will be trapped within this 10 picoliter size rather than 10 microliter size. So the local concentration of DNA or RNA will be much higher to the level that you can do direct detection without amplification. So here is the concept. So you can use this for focusing device to generate millions of droplets. And then what we do is like we mix the sample with the molecular beacon, is another freight dice, and then bind to the 16S RNA. So when the droplet contains the bacteria, you will release RNA and will be bind, and we can see the color change. Initially, you have a tracer dye that show the droplet, but if you see the different colors in the droplet, you know that this droplet contains bacteria. The other droplet does not. This is a digital way you can achieve a highly sensitive detection, but you can also do very precise quantification because you detect and quantify bacteria one at a time. And this is the result showing that you can device here. You can show with this one, you will be able to show this is the digital result here. You can see here you are able to, like these are 4 droplets. This actually is negative control. You don't see any drop, but this positive control, every single droplet you see the signal. This real experiment, you can see, some droplets does contain one bacteria, these 2, and this middle one does not. So you don't see this high peak of the red signal here. So this is a way of digital detection without the use of PCR amplification. I'm going to talk about quickly on the second droplet platforms that we are interested in. We have been working on is this the open surface droplets. So here there are 5 droplets of identical volumes, but their shape is different because we adjust the surface energy. We create a surface energy gradients on the substrate. What we did is that we create a micro post, and we can contain the micro post to adjust the surface energy. Then we are able to adjust the contact, change the contact angle, and then change the shape of the droplets. The next few slides, I'm going to share with you that we can use this surface topology together with magnetic particle to manipulate droplets, allowing us to do sample preparation and also do detections on a single device. So here is that a lot of people here, you are doing PCR, you are doing similar projects. It's microfluidic to do sample preparation of PCR. So this one, you know this concept that PCR require a lot of sample preparation steps. You sterilize and DNA binding, wash, elution, and then you send to these PR machines here. So what I'm going to present to you in the next few slides is that we use this microfluidic droplets platform to carry out the entire process. And we first use the droplet to serve as storage, to store all the necessary chemical or biological reagents used in the entire PCR process. The first droplets are stored with a lysis buffer together with a silica magnetic particle to capture the DNA. And these middle 3 droplets here are the storage of the washing buffers. And the final one is the emulsion buffer together with the PCR buffer. Then we are going to extract the DNA from the sample droplet all the way to the final droplet and do the PCR. So I have a, before I show you the results, I'm going to show the basic concepts of this assays that we use a silica magnetic particle to serve 2 purposes. For one is that we are going to using silica magnetic particle. They will be driven by magnetic fields. And then the particle will drive the aqueous droplets to fuse, to merge with the other droplets for mixing. And then you can also split the magnetic particle from the aqueous droplets. And this particle carries DNA. You can send this particle to the downstream droplets for another mixing or reactions. The second is that, again, the particle is coated with silica, so we can use it to facilitate side effects extraction through the silica DNA, silica and nucleic acid interactions. So I'm going to show you this droplets here is that, again, you have a couple droplets. First droplets is 6 droplets. The number 1 is the lysis buffer droplets. And these 3 are buffers droplets. The final one is the elution and also PCR reagent droplets. So what we did is that we dispensed the samples, can be blood samples, directly to mix with the first droplets. And then we will extract DNA and guide the DNA all the way to the ends, and we do PCR directly on the device. So I hope this video works. It does work. So you dispense the sample first to the droplets here, and then cell-organized DNA will be captured by many particles here cluster. And you move this to wash buffer 1, 2, and 3. You can use only 1, 2, 3, or even 4, depends on what reagents, what kits you use from individually from different companies, depends on your choice. And then the purified DNA subjects will be sent to the downstream, and then you will be able to do PCR directly in the droplets. And you have we have a miniaturized optical detector, fluorescent detector, right on top of it to monitor the signal. So with this, we can do the sample preparation and detection directly within these droplets, because everything is filled with oil, so we can do emulsion PCR, because purified DNA is in the end. So we can monitor the PCR amplification within these droplets and monitor this is ISF1 gene, this whole plot, monitor this ISF1, be able to monitor the real-time amplification of PCR using this technique. Okay, so the final last few slides I'm going to show you is that the previous slides I show you is that we guide the transport of a magnetic particle using permanent magnets. In fact, it was done manually. Still use it. You can use the transition stage to control it. But I'm going to show you, you can also use electromagnets to control the particles. So here is very straightforward. You can use a printed circuit board, PC board, very tiny, very small one. Here we print 2 layers of electric coils. You can make a coil. If you apply current here, you generate magnetic fields. You can guide the transfer of particles. So in this case, that if you can generate 2 different layers of coils, this cross-sectional view, and you can apply currents of a different polarity, you will be able to generate electric fields, high field and low fields. You know the particle will be driven to the high fields. So with this, you can use the coil without the use of permanent magnets to guide the particle. In this case, you will see this droplets is driven by simply applied by the currents to these coils on these printed circuit boards. And then you can also use this to do splitting. You can, using the field, try to split the DNA containing particle from lysis buffers to fuse to these wash buffers and already go down. And then you can also use this one to do, mixing is tricky and important. And you can also control to max the field to be maxima to be the center and switch the polarity to make it to be field minima. By doing this, you will be able to perturb and agitate the particles to facilitate these mixings within the droplet to enhance the kinetics. And then And then, so here, again, I'm going to show you here is different version is using electromechanism. So showing that you are, I hope this works. Okay. So you are able to, this is actually entire DNA separation and also binding. So this is the binding buffers here. So you see you will be able to do the whole thing. You capture the DNA and go through the first wash buffer and then code and then the second one. You will see there's some residual compounds there. So this is why we need to use multiple wash. Depends on the kits you used. And then gradually become much cleaner. And then the DNA in the end will be driven to the downstream and DNA will be eluted in the end. And you can also do the same thing, do the real-time monitoring of this PCR using this device. Okay, so basically this is what I am going to share with you. So in the end, before I conclude my talk, I would like to send the students contribute to certainly this is all done by the students and fellows. But mainly I would like to thank a lot of collaborators at Hopkins here. So we have been working with many medical scientists at Hopkins. And funding-wise, my major fundings, big part of funding is from NIH, but also have some funding from Center for Cancer and also STARPA and also NSF. So with the list, I really thanks for your attention, and so I will be very happy to answer any question you may have.
All right, any questions?
Yeah.
So on the, when we first started talking about the Droplet Microfluidics, we're showing the non-starving active single sequence effective. Were you bringing in the whole cell and doing it, or taking isolated genomes?
Whole cell. Yeah.
So you actually have to lyse the cell?
Yes. Yes. We have to lyse the cell to do it. So the one is not. If you do whole genome, it's become digital PCR. So this is not digital PCR, it's single cell. So it's, oops, I have this project, sorry. Can I do this? So in fact, I should show you the device here. We do the digitize this sample, direct the bacteria samples, and then you encapsulate into a droplet, and you will go through these lysis regions. You look at here, you have this heater. Basically, we do thermolysis. Sometimes depends on bacteria. If E. coli, no problem, you can do thermolysis. Some bacteria you cannot lyse. You need to combine also with chemical reagents there. But time is very important. Make sure before they get lysed, it's already digitized within the droplets. But for this E. coli, you can just use thermolysis, and you can lyse. And also in this incubation regions, the probe also binds to a resisting sRNA, so you do the detection. So this, the result I showed to you is begin with the raw samples, intact cells, and lysis is take place on chip.
Have you tried any single mammalian cell analysis?
We have not tried single mammalian cell for this project. This is mainly for infectious disease. And mammalian cells, it will work, but I have never tried. But droplet has to be much larger because this droplet is only about 10 microliters size. In order to do mammalian cells, you need to use larger droplets. And larger droplets, on one hand, is like the ultimate concentration will be lower than what you would like to see because smaller one you can fully concentrate. But in principle, it will also work, but we have not tried yet.
Do you recall if the underlying rule, like say gram negative versus gram positive, one will light under sort of the more complex, which is very.
We have not tried many. In fact, this paper only show the E. coli. But other devices we have shown that thermal definitely is not sufficient to lysis a lot of gram positive, even gram negative is difficult to lysis. So a lot of this is for E. coli. But if you want to demonstrate different things, likely you have to couple with other enzyme, protease K or chemical reagents to your droplets for the more tough bacteria with tough membranes. Yeah.
Okay, one question.
Yes. What is that? Oh, sure. Yeah, that's very good. This good. Electroweighting, what you see, some people may confuse, looks like electroweighting. You can do something here. But electroweighting can only manipulate droplets, cannot do assay. So here, the unique part is we combine solid phase extraction. The magnetic particle is not used to just to guide the droplet to fusion. We do reaction to solid phase to extract the DNA to do this one. Yes, yeah, in fact, yeah, that company already do it. So that's what, people have done using electroweighting EVA to do something similar to this. They have to add magnetic particle to their solution. They have to add an additional magnet to their system to pull out the particle, the guided one. So they can do the similar thing here, but it's different from what if you have to use permanent magnets to do the whole thing, your system become more complex. What we see is why we have to use electroweighting. We combine just magnetic field to do the two purposes, not separated and using two different actuation mechanisms. Yeah. Yeah. That's right. Yeah. 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 Well, that is right. But the geometry, in fact, I can show you another. I don't have time. To have time, I can pull out one paper, or I just quickly. You can. We have a paper just published. Yeah. 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 So basically, geometry may not be necessary. So we have a paper just published actually, let's say in Advanced Materials. Is we call sets, basically using Teflon coating, and then you can using that one to manipulate. So you don't use the one. But I agree with what you say here, but I have not finished what I just said. It is a waiting. There's one fundamental limitation for this kind of application because the volume is so small. It is a waiting, especially EY. The volume, you can see the droplet is very small. If you're talking about clinical sample, I have a lot of people, even from Canon here, I saw here. You really talk about infectious disease, real samples. We're talking about beginning volume. You're talking about it's not 0.5 microliter. You're talking about 20 microliter or maybe 100 microliter to begin with. EWA just cannot handle these big volumes. I should be careful. They can do, but typically this is the volume is very limited, not in the micro or tens of microliter region. So there could be a potential issue for using EWA to do real assay. Depends on your thing. So this kind of general comparison. They are all pro and cons, right? So just general comparison to answer your first questions.
Other questions?
Yes. Yes.
You've obviously done very well over the last decade at getting access to patient samples and collaborating with data, something in the better culture at Hopkins. That's something that you've had to work very hard at. And oftentimes, yeah.
Right. Well, so I actually shared a very same topic with Wang just now earlier. So it was, I guess, it's part of a culture of Hopkins, but not necessarily just culture means that you go there and then it just happens. It's not necessarily there. So I did spend a lot of time when I was juniors and building up this trust with collaborators. Otherwise, like we shared, oftentimes in the very beginning, it's like you have very exciting meeting one or twice, maybe 3 times, and then no follow up because everyone's busy, right? So you got to build this one, and then we did spend and location is really an issue because we are quite close. We are only 15 minutes difference, so we can see our student can come to different labs. So we have this one, and gradually when I start to do this one, the people trust, okay, you are not only interested in get some idea, find some name from this department sample and you publish it, they go away. Eventually, with the sub-problems. And they know actually I'm really interested, they don't really care about your forensic technology. They just want to make sure you can solve my problem. Right, so if you trust that you are alone, so different people have different agenda. My brain go to different statistician than statistician. I know what this statistician like. But I let me make it clear that it's whole thing we can work together for center for different things. So I mean, it's several years that we realize the trust. So right now it's like I would say partially it's really environments that Hopkins has is we're closer, and then we have some successful collaborations of this kind before. So people know, yeah, it can work. The other thing is like personally in my lab and some other PIs interested in translational studies, investing a lot of time over the past years. People know, yes, this guy is the one not only in just device, but he really understand and motivated to do medicine. So, yeah. Yeah. Mm-hmm. You're talking about which disease and talking about which application? Yes. Right. Right. Yeah. Well, I understand. I guess I can try to understand and answer your question. First of all, the DNA integrity is not a specific detection. You never using DNA integrity say this is lung cancer, this is cancer, because that one does not tell you specific. That's what I mean. It's general screening. That's good for monitoring or monitoring the tumor burdens, tumor size. It's a very good one. If you want to do diagnosis, certainly you cannot measure size because size does not give you genetic sequence here. But you can couple that one with other sequence. I show methylation on promoter gene different thing, mutation, right? It is very, very one. So they and how that's why I mentioned that genetic detection is being very effective. But you say they are all, for example, block 1, block 2 mutation, you know it, and also different thing. They are something has showing the drug resistance, right? But how widely is it used? Certainly is not widely used. That's what a lot of research still ongoing here. But there are a lot of, for example, we show that you can detect. I can tell you that we published one paper last year in cancer research. I mean, pancreatic cancers. That shows that we can detect 90%. There's nothing 100% sensitive and specific. You cannot get this one. In the future, maybe, but it's very difficult. So it means there's no marker. It's really the magic that you detect positive this disease and negative there's no disease. Always you are talking about 80%, 70% versus 90% or 95%. There's no 100 sensitivity specificity, I want to tell you. However, there are a lot of markers being shown for specific cancer or specific stage or companion diagnostic that has highly correlation, sensitivity specificity up to 95% or 99% or 90%. There are a lot of examples of this kind. But overall, you can say it's difficult to say what is the general, what is the level of success of circulating DNA for cancer or for other things. What is the overall success? It's difficult to define this, but depends on what sensor, depends on what kind of a treatment you are looking at. There are some very good successful examples. Mm-hmm. They are. I can tell you, even for cancer, how can you do cancer detection? Early cancer detection. You say imaging is right now the most reliable. When people see how they diagnose cancer, they see something tumor, and even you see something, you cannot make sure it's tumor. You got to do tissue biopsy, make sure, yeah, this is tumor tissues. You think about this. When you can see tumor, it's already centimeter size or larger for you to see it. Certainly, people are fighting to see something you can detect at early stage. How? Marker is one thing to do it. And you can talk about protein marker, DNA marker is different thing. There are pros and cons here. Protein is easy to do, but DNA may be more sensitive and maybe can be captured earlier. So this is kind of these routes, but how overwhelming this success, how successful it is right now. Certainly, it's under what I mentioned, there are a lot of limitations. But you answer me like, why doctor, they love to have something, holy grail, if they have something that can show you get one blood, you look at DNA or protein, nonsense DNA. They can tell early stage, very high specificity. So this is what a lot of NCI still putting a lot of money on this gene. You have a circulating tumor cell is one thing, they look at the tumor cell. still, in the end, you got to go back to see what is the genetic or epigenetic or the protein profile from the circulating tumor cells. So we are doing is that they look at the circulating DNA to see the whole thing, to see whether we can find some marker, we can correlate better with specific tumors, and maybe with some companion diagnostic, all different stages, right? So this kind of general direction, certainly we cannot claim it's a great success, but this is very promising, we believe, direction to go for.
All right, so let's take that's a long one.
Thank you. Yeah.